welcome to a Biomedical Science Society podcast. This week we have Dr Victor Bolanos-Garcia, a senior lecturer at Brookes University for over 8 years. He is a lecturer in multiple modules, but you are likely to have him in any biochemistry subjects. For example, he was a module leader throughout our first year for Introduction to Biochemistry A and B. Dr Bolanos-Garcia graduated with a bachelor's degree in pharmaceutical chemistry, a master's in molecular biology, and a doctorate in biochemistry, all from the National Autonomous University of Mexico. He moved to the UK to study the mechanisms of regulation of cell proliferation and motility by MET kinase receptors with Professor Emmanuel Gerardi at the Laboratory of Molecular Biology of the Medical Research Council in Cambridge, and subsequently to study the structural basis of DNA repair mechanisms and other complex signaling networks with Sir Tom Blundell at the Department of Biochemistry, University of Cambridge. His research throughout his master's, doctoral and postdoctoral qualifications led to his current studies on mitosis and the Spindle Assembly Complex checkpoint, or SAC. Thank you so much for being with us today, Dr. Bolanos Garcia.
Thank you very much for the invitation.
We're very happy to have you here, and we're very excited to then be talking about your very, very interesting research. So thank you again.
Thank you for having me here today. Yes, I'm very excited about this.
Yes.
Okay, so I'll start off with the first question. So you completed your bachelor degree in pharmacology chemistry, which led you to later pursue a career in molecular biology and ultimately your PhD in biochemistry. Could you tell us what your research was about during your doctoral research?
Yeah, sure. My doctoral studies aimed to understand the relationship between the structure and function of a family of proteins, human proteins, that mediate lipid metabolism. So essentially, I was trying to understand how the physical structural details of the molecules that underpin lipid metabolism in the human might pave the way to enhance our understanding of the function of those molecules. Particularly the molecules that were involved, proteins that were involved in the metabolism of cholesterol. But my master's degree in molecular biology, the training I received there, was really instrumental to incorporate molecular biology approaches, such as molecular cloning and the production of the recombinant proteins in bacterial cells, to the study of human lipid binding proteins. So yeah, and my PhD project went well, led to the publication of 8 of 9 papers. And I was awarded a nationwide prize, the Weizmann Prize for the best PhD thesis in natural sciences in that year. So
wow, that's amazing.
Amazing, gosh, for 9 papers, that's such, and all within similar context. Yeah, productive.
Yes, yes, all of them about the the structural understanding of these lipid binding proteins, and also got training in protein crystallography. So by the time I started my master, which was about molecular biology, and it was the project was about the production of a recombinant enzyme in a gram-positive bacteria. At that point in time, I realized that having a structural understanding of the molecules that perform important activities in the cell, as well as the design of molecules that are of industrial interest, really require a deep understanding of the structural basis of the molecule, the structure of those molecules. So that's why I started to gain very much. I was very much interested in the study of the relationship between protein structure and function. My PhD allowed me to explore those areas, and alongside the study of protein molecules, the production of the molecules in a recombinant system, the purification of the proteins from the blood, from blood. I was at the beginning of my PhD project. I was visiting hospitals every 2 or 3 weeks, collecting samples of outdated blood samples from which I isolated these proteins. It was really, really hard work to isolate these because they are present in minor concentrations in the bloodstream. So that then brought me to investigate more about the production of these proteins in a recombinant system. And alongside, once got enough material to study the biochemical aspects, I mean, physical aspects of the interaction between these proteins and lipids, that also paved the way to attempt to crystallize proteins and so the structure of these molecules using X-ray protein crystallography. So yeah, and that it was successful because I have papers that dealt with the new methods to crystallize proteins, as well as all the studies regarding the biophysical characterization of human lipid binding proteins, which at the time were the way they bind lipids and how they interact with each other and are regulated was poorly understood.
And was crystallography a very new process at the time in terms of its application with studying protein function and the structure, or was it already starting to emerge in the field and there were more and more cases and more investigations using the technique?
No, in fact, I was one of the very first in Mexico that actually got the training to do protein crystallography. It was really new in the country. It's a professor, Manuel Soriano Garcia, a very well-known professor in Mexico. He spent many years working in the USA. He worked in Buffalo, worked with Nobel Prize winners.
Wow.
And then he moved back, and for Merck, his company, and then he moved back to Mexico and started his group of structural biology, and really structural biology in Mexico started with him. I was very lucky to be one of his very first PhD students.
So taught by the best, yeah.
Indeed, it was really amazing. We have an opportunity with him. He's a very supportive mentor, a great mentor, a great scientist. I had the opportunity to attend international conferences. I remember one in Seattle. It was the 100th anniversary of the discovery of X-rays. And there were a large number of Nobel Prize winners attending the conference. I have pictures of myself with a number of these remarkable individuals. And Professor Soriano knew some of these guys, so actually he invited some of these Nobel laureates to Mexico. And it was funny to see these lectures, very formal setting with these individuals. And in the afternoon, we all went for tacos and have a very formal chatting. Yeah, it was a great time.
Yeah, another different side to the scientific community, which I think from the outside, like you said, it seems like a very, very formal and very diplomatic kind of environment. But instead, there's always we're all humans at the end of the day, and we're all going towards a similar goal.
Oh yes, and very, very supportive individuals. I remember having this lunch with Johann Eisenhower. He was awarded Nobel Prize with Robert Hoover and Helmut Michel because of the first structure of a membrane protein, which was a revolution in the field because it was the dogma at the time that was you could crystallize every protein you can think of, but membrane proteins. And they proved that, you can crystallize membrane proteins. And it was the first, bacteriosin was the first protein to be crystallized, and they were awarded Nobel Prize, and rightly so, for this really major breakthrough in the study of protein structure.
Gosh, what resilience. Could you share with us the thought process and what was going through your mind behind moving your studies from being in Mexico and living in Mexico your entire life all the way across here to England, and what kind of happened? Yeah.
Well, as I said, I was very much interested in structural biology right at the start of right in the middle of my master training. So I knew that I wanted to do structural biology, and my PhD project allowed me to start exploring. And I was also lucky that the University of Mexico was supporting PhD students to attend overseas conferences. And it was one in Cambridge, in which I presented some work, and I was presenting a poster, and next to me was a postdoctoral fellow, and he started chatting from India. And he started chatting and say, "Oh, where are you?" Well, I'm working in Cambridge. I work with Professor Tom Bundell, who is one very, very prominent scientist in the field of structural biology. I mean, it is a classic textbook written by him and Louis Johnson. It's a textbook on SD textbook still on protein crystallography. So, and he said, well, he's actually attending the conference. He's going to come and have a look at the posters and talk to people. So, well, okay. And then, well, one of the things is that I wasn't afraid of talking to him. And when he showed up and said, "Oh, are you Tom Bundell?" "Yes, I am." "And can I show you my poster?" And he was like, and I well, in my broken English at the time, I was explaining my poster, and he was kind of, he was very patient, and they listened to my explanation of the poster I was presenting. It was about a bit molecular modeling of a living binding protein, by the way. Of course, and then I just asked the question, I'm doing my PhD, I'm really interested in structural biology. I know you are one of the big players in this field. What are the chances of doing some postdoctoral training in your group? And I think he was a bit, I was a bit naive maybe, but he was kind of supporting and said, well, you want to come along and visit the lab and see what you think. So he made this, he's a very busy man. And he, in his agenda, he booked some time for to show me his lab. And of course, I was really impressed by the infrastructure, the quality and ambition and scope of the projects. And I just kept in touch. He said, well, as a non-European citizen at the time, it was really difficult to get any funding to come, from Mexico to do any postgraduate training. So I had to wait 1 year until the government of Mexico launched a call for, it's a competition for scholarships to do postdoctoral training overseas. And and I was lucky, I was awarded one of those. Then I started my work my training in crystallography in one of the best groups in the world in Cambridge. And then once success led to another one. So once I was there, my plan was to stay just for 1 year. Then I got a renewal of my scholarship for a second year. Right in the middle of the second year, was invited to apply for a prestigious scholarship, and I got it. It was a international research fellowship from the Wellcome Trust. And so I stayed there for another 3 years. And after that, Professor Blondell was awarded a program grant, 5 years for funding, substantial funding, again by the Wellcome Trust. And he invited me to stay with him for another 5 years. And then we applied for another grant together to Cancer Research UK. And we were lucky enough to get the grant and so on and so forth. And at some point, I knew it was time to move on and start an independent career. So, and I was very lucky that the Oxford Brookes considered my application for a senior lectureship, and then I was appointed. And yeah, so that's and when the Professor Blondell, I was studying mechanisms of DNA repair and a structural understanding of how the DNA is repaired after being damaged by X-ray radiation and also one of the sources of radiation. So I was very interested about this problem. But it was a very busy area. It was a very competitive area. Other groups around the world are still working on this very same problem. And something that caught my attention was the fact that when the DNA is damaged, there is a number of systems that the cell can resort to in order to repair the damage to DNA. But one of the features that I found really interesting is that in order to give the cell a chance to repair the damage caused to the DNA by gamma radiation, for example, the cell cycle has to slow down a bit. So cell division has to slow down in order for the cells to repair the damage before they commit to division. And I was like, oh, that's pretty interesting. How they do that? And then I started to investigate about this and found that it was very little known about the actual process.
On how it slows down.
Or how the cell cycle is slowed down. And then I came across this fascinating mechanism of the Spindle Assembly checkpoint as being responsible of that, monitoring the proper attachments of the chromosomes to microtubules. And when the attachment is effective, the Spindle Assembly checkpoint is active. And again, the end result of the activation of the Spindle Assembly checkpoint is the delay in a mitosis progression, again, allowing the cells to repair the damage. Or in this case, the misattachment of the chromosomes to microtubules. Yeah, that's something that I found really, really interesting. And then very little was known about the actual process, let alone about the structural understanding of the process. And so I started to work on this problem and keep working on this problem ever since.
And when
Amazing.
did you start this project? So since your time you started at Brookes, or was it already at the Blundell?
No, I was in Cambridge again.
Already in Cambridge. Yeah.
Blundell, yeah, he also Tom Blundell is a great mentor. So he allowed me to, alongside the work I was appointed to do, which was all about the repair mechanisms. I was also supervising PhD students, I provided training to undergrad students.
Gosh.
At the same time, I was given a chance to start exploring these ideas. And yeah, we have some nice publications together as a result of this exploration of spinal checkpoint with the Blundell lab.
And I mean, like, I think Oxford, England, we're all very multicultural. There's a lot of people that speak 1, 2, 3, plenty of languages. And I wondered, like, me myself, I'm bilingual, I speak Spanish, but I don't really know where to go in order to be able to. So I can speak Spanish, I can go to Mexico, Spain, and have conversations, get around, get to know people. But I think learning the Spanish words for scientific things that I know in English is really tough. I have a really hard time. Like, I know some of your papers were in Spanish and I was trying to read them, and it's almost like I have to Google 101 things because I just don't know those words in Spanish. I know every other word, but not the science words. And I wonder really what your advice would be on people, who are like that, who maybe are bilingual or whatever, and they're looking to expand their knowledge in terms of maybe more of a scientific aspect and open up their avenues of the papers they can read and things, because I feel like it would be really, really beneficial in terms of like how did you manage it?
Well, I knew that the national language in science is English, as a matter of fact. So at any conferences, I don't know, it was an eye-opening experience because I knew how important it was to be able to communicate in another language that wasn't my mother language. And then it's essential really to communicate with the people to learn English. And if you're interested in languages other than English, then you should have also real interest in the cultures. I mean, Spanish world is a very diverse and vast. Spans all Latin America, Spain, and other places around the world. So learning Spanish is, it's also another way to look at the world with different eyes and expand your horizons, understand other ways of thinking and feeling as well. Because we Latin people are, very passionate and outspoken, and in English, at least in Britain, it's it's a different way socially. So yeah, you can also open other opportunities. And Luca can tell you about that because he's also
I'm also
Latin person, so in Italian. So
bilingual
you know what I mean by passionate,
absolutely
outspoken.
Yeah. And I was going to ask, Doctor, I mean, now, very fortunately, with, I think, globalization and how widespread English is now, of course, in the scientific community within the last 2 centuries, really. Do you still believe there is, like what Priscilla was saying, a bit of a language barrier where perhaps some very good scientific investigations and reports and articles aren't getting as much recognition or, how can I put it? Yeah, recognition or not as well known because they're in a different language. I mean, I haven't really personally looked if there's a lot of Italian researchers, but I think like especially the Spanish spoken world is much more vast than the Italian. I think, unfortunately, Italy's the only country which actually speaks Italian is actually Italy. It's not Spanish, where a number of countries speak the language. Do you still think that a lot of papers could be recognized if English would be much more more widely spread? And actually, if, like what Priscilla was saying, that if students, instead of just looking at the English papers, but they will go beyond. I mean, me and Priscilla are very lucky in being bilingual, but maybe to those students who have this, I guess, gift really of having another language, should go and see other papers in the other tongues and in other languages.
Yes, and I guess this is more sort of important perhaps in the humanities and the social sciences, where you are really trying to understand the realities of a particular community in the Spanish speaking world. you really have to, go deep inside, try to understand the way people feel and behave socially in that community. In the natural sciences, perhaps it's not the same. It's the same pressure. But we know when you do your research, you want to disseminate your work the best possible way, right? The best possible ways. And so we know that the vast majority of scientists read certain type of journals. So you aim to publish your results there because, it's, you ensure that you have, a wide readership being aware of your research, and then really open up opportunities for collaboration and, so it's a different type of pressure. What is fair to say, and because I have heard that a few times, is that there is a sort of I don't know, it's called unconscious bias. I don't know how unconscious that bias is. That sometimes, because I've seen this, sometimes you see papers that come from a particular, part of the world, and the way you look at that paper is not the same as it was coming from Oxford, England.
The style of it, or the approach to it more.
Yeah, even the, I mean, I've seen this, I have first-hand experience on this, that the way they examine your paper is not the same. They're really looking for minor things to tone it down. And sometimes that's not, I mean, sometimes you see this as a bias. Sometimes you see, instead of having a revision that aims to help you to convey your message in a better way, or even enhance the quality of your research work, is trying just to find ways to not to publish it. And sometimes I've seen that it's as a result of sort of a bias, like this just having this preconception that, oh, this is coming from Mexico, it's not going to be great science, it's not going to be cutting-edge science. Which might not be true. I mean, there are a number of pretty, world-class groups doing research in Mexico. I mean, that's just as an example, but I've seen that sort of bias a number of times. And I felt it more when I was in Mexico, when I was preparing my own papers. But I've also seen that with other colleagues, because I also sometimes collaborate with people in Mexico. And sometimes they ask me for a second opinion about a manuscript, and I see the paper, read the paper, say, well, it's pretty, solid story. It's well supported. The science is sound. The problem is relevant. You shouldn't have any problem in polishing this. And then you see the referee's comments or the review's comments, and you see they're just going through
the minute details,
read it, trying to undermine the paper instead of being constructive. And that's paper coming from the Cambridge, because I have papers that I wrote when I was in Mexico. Papers I wrote when I was in Cambridge. Pretty much, I mean, the same, training, the same way of looking at my problem. I'm the same person, and still the way the papers are, welcomed by the journals was different. So. But having said that, I mean, there is a lot of ways to communicate your research now. You have even online journals, and attending conferences, organizing virtual conferences these days. So there are ways in which you can really make your work be known worldwide. But still, there is a sort of a barrier.
Fantastic. No, very interesting about the approach and on, I guess, examining different papers and reports. Going back to your own research, and currently you are developing your research on the Spindle Assembly Complex, what you were saying, which is what slows down the cell cycles when DNA is damaged, and which is involved in a series of complex signaling pathways, and they are essential in the transition from metaphase to the anaphase division during cell division. Could you tell us a bit more about this complex and what the actual aims of your research? I mean, you briefly mentioned before, but maybe go into more detail now, what the aims of your research are, and even in terms of developing eventual anti-cancer drugs, and how would the basis of the drug development work in terms of the research conducted? So how would the stages from your research can help in the drug development?
Yeah, I mean, the overarching theme is that the molecular understanding of the mechanisms of cell division regulation remains a hugely important problem in life sciences, and the SAC, the spinal checkpoint, plays a fundamental role in ensuring genome stability. So at the end of the day, when the cells divide, they pass on all the genetic information to the descendant cells. And the spinal checkpoint is a mechanism that ensures that the genetic material is passed on to the descendant cells with the high accuracy and fidelity that is required in order for the species to prevail. At the end of the day, that's the ultimate goal for the species, is to self-perpetuate. And for that, the maintenance of a stable genome is critical. So the spindle assembly checkpoint plays this fundamental role. And it's fascinating because being so important to ensure genome stability and definition of the genetic material with high fidelity, it's not surprising that it's tightly regulated. And there are around 100 different proteins participating in this signaling system. So it's a very complex one. And of course, all these events, crystallography of protein, DNA, protein-protein interactions that are remodeled in the space and time. So the regulation of all these interactions and how these interactions happen in the first place at the right time, in the right place, is truly fascinating. We still understand some of the aspects in great detail, but there's other fundamental aspects about this process that remain obscure. It's a fundamental process in eukaryotic organisms, so it's highly conserved across species, from the fruit fly and the humble worm C. elegans to body inefficient yeast to rodents and mammals. So, but the devil is in the details, as I said. So some interesting aspects, you're also interested in the evolution of complex mechanisms of cell signaling, such as the SAC. You will see slight differences in terms of the relative importance of some of these proteins in one species. They might play a fundamental role in some species, but not so important in other species. And these differences are also one window to understand the evolution of complex signaling systems and, of course, evolution of complex organisms. So it has really, it's a system that is so intricate, complex, that you can start studying this system from various standpoints and still be fascinated about it. And one of the key aspects of how it relates to the clinic is the fact that the effects in the SAC are the effects in the SAC are the prime cause of spontaneous abortions. Also lead to the effects in this process of SAC signaling, lead to developmental defects. Down syndrome, of course, is Down syndrome is caused by this uneven distribution of chromosomes in the cells, leading to trisomy 21. Cancer, all aggressive forms of cancer are characterized by the presence of by the harboring an unstable genome, and that means you have uneven numbers of chromosomes in cancer cells. So the SAC is at the heart of these problems. So understanding how the system works under normal conditions is important for a fundamental, it's a fundamental molecular question, but also understanding how it works in normal conditions and what are the reasons it can go wrong and lead to disease, of course, provides an opportunity to develop novel therapeutics in order for diagnosis, as well as for the treatment of diseases such as cancer. And so my research has this other, there are two main elements of research in my group. One is the basic sort of research question, trying to understand how the spindle assembly checkpoint actually works, how it is regulated, how these complexes are how these complexes are formed in the cell at one point in time, how the positional complexes, when the job is done by one of the complexes, lead to the formation of other assemblies, with the ultimate goal of ensuring the accurate segregation of chromosomes prior to cell division. And the second aspect of research, which is more sort of oriented towards the traditional research, is identify the proteins that are associated with diseases such as cancer and developing strategies to interfere with this abnormal SAC signaling in cancer cells. The approach I'm using at the moment is the development of a small molecule that interfere with mitosis progression in cancer cells. Wow. And we have some success on this along those lines. We have developed a number of molecules that have proven to be effective in the context of breast cancer. And we just applied for a European Union application, patent application recently, and we're in the process of filing a second patent application to interfere with the abnormal SAC signaling in cancer cells.